Thank you for tuning in. The work that I'm going to tell you about is a large collaboration in which we're looking throughout the fly brain to ask what the spatial and temporal scales of brain-wide neural activity are as a fly executes simple behaviors. So thinking very mechanistically, one can ask what the minimal circuit is for a given task. We know that small circuits can drive behavior, so in the simplest case, a few synapses can get you from sensory input to motor output. This way of thinking has proven very useful. But we also know that there can be a lot of structure in the activity of populations in a brain area. So in mammals, for example, it's becoming more common to think about the dynamics of a network driving behavior. In this case, the identity of the neurons is not important, but rather each neuron is just part of the large network and the global pattern is what matters. Moreover, even at the level of the whole brain, there are meaningful dynamics. So in this example from the Churchland lab, you can see waves of activity across the dorsal surface of a mouse brain. These observations at different scales lead to conceptually very different ways of thinking about how the brain works. The problem is that this is usually also coupled to methodology. What you see depends on how you look. To see these scales simultaneously is challenging. We chose to approach this problem in the fly, where we know a lot about single neurons and we have access to the whole brain. We used SCAPE microscopy to image a large fraction of the fly brain at 10 brain volumes per second while we monitored the fly's behavior in a ball. We expressed GCAMP6S and DS-RED, both localized to the nucleus. So here, color indicates depth in the third dimension, and the cartoon on the left shows roughly what you're looking at. We're imaging from the dorsal surface, and the cell bodies form a heart-shaped structure, with black areas in between being neuropil. Now looking at all three dimensions, you can see rich dynamics in both space and time. So when we look at the activity of every neuron, the first thing that's apparent is that virtually all visible neurons are active. These are all the neurons we can see based on a static marker independent of activity, so we might have expected to see many neurons doing nothing, but we don't. Not only are most neurons doing something, but you see strikingly that most neurons are doing essentially the same thing. So to understand what we're looking at, our approach was first to look at how much of brain-wide activity we can explain from behavior, and then to dig into what's left over. So when we look at the corresponding behavior of this fly, you see that the green state, which shows bouts of running, aligns with the activity of most neurons. So the activity of most neurons correlates with running. We then fit a simple regression model using all behaviors to explain the activity of each cell, and we allowed for behavior to additionally be smoothed and shifted in time to fit each cell. So you could see from the raw raster that most cells track running, but what wasn't obvious, but is revealed by this approach, is that there's a broad distribution of the time scale with which neurons track behavior. So here, cells are colored according to their behavior time constant, and you can see that cells with similar time constant co-localize. The clusters of yellow are particularly interesting because a large time constant means that a cell tracks a smoothly varying estimate of behavior. So yellow cells encode the tendency to run rather than instantaneous running. This might correspond to something like an arousal state. The next question that we asked is how much additional structure there is in brain-wide activity that isn't explained by behavior. So after behavior-related activity has been regressed out, the residual activity exhibits rich dynamics across both space and time. Here you're looking at a sample of 100 neurons sorted by similarity, and you can see these really rich dynamics. When we do PCA on these residual dynamics, we find that the activity is high-dimensional. These patterns, or PCA modes, tend to be very sparse, on average involving less than 20 neurons and sometimes involving as few as 4. Really strikingly, many of these modes are not only sparse but spatially organized. So here is an example where a pair of cells in each hemisphere have highly similar dynamics. So this formed one of the dominant modes of activity in the whole brain despite basically only being comprised of 4 cells. These patterns are also common across flies. So this suggests that there are a large number of small patterns, clusters of neurons with unique dynamics. We asked this directly using hierarchical clustering. This analysis revealed interesting structure at many scales, but focusing on the small clusters, we find that they too display spatial symmetry. Here we're seeing two cells on opposite sides of the brain whose activity is more similar to one another than to any other cell in the whole brain. This is one example, but this general phenomenon is true across clusters and across flies. So these observations are remarkably consistent with what we know about genetically defined cell types in the fly brain. There are of order 1,000 genetically defined clusters of 1 to 10 neurons in each hemibrain, so our data are consistent with the expectation that these cell types have functional consequences. Finally, we've found that small clusters participate in the brain-wide dynamics. So here you can see that the distribution of running correlations for cells that form a cross-validated two-cell cluster is indistinguishable from the distribution over all cells. We've dug into this quite a bit, but the bottom line is that small clusters appear to also participate in the brain-wide dynamics. So in summary, almost all cells are active when the fly runs. A lot of neural activity is not accounted for by behavior. Activity across the brain is high-dimensional, and most of these dimensions include very few neurons. The same neurons participate in brain-wide and localized dynamics. So neural activity in the fly brain is organized over multiple spatial scales. There are a large number of small clusters that are likely genetically specified cell types with functional consequences, but there are also large-scale dynamics in the fly brain. So two conceptually different ways of thinking about the brain, microcircuits and manifolds, are both relevant, and they explain different aspects of activity in the same brain. So what this suggests is that microcircuits operate in the context of global dynamics, which is probably itself shaped by the operation of other microcircuits. So I'll end by thanking everyone who is involved and our funding sources, and of course thank you for tuning in.